I'm here with Dr. Mahesh Kote. He is a postdoctoral researcher at the University of Concepcion in Chile since 2018. Currently, he is conducting research on a combination approach of statistical optimization and mutagenesis for improved production of lipids as biodiesel feedstock from a newly isolated yeast strain that originates in the Chilean rainforest. So he's going to tell us a little bit about that today. And thank you again for joining us for this webinar. Should you have any questions or need assistance, please use the question or chat box. At the end of the presentation, we will open for questions and get those answered, but please feel free to write those in at any time. And with that, I will turn it over to Dr. Kote.
Okay, thank you, Amy. Hello, everybody. Greetings from Chile, the other end of the world. I thank AOCS, first of all, to give me this opportunity. This is also the first webinar for me, and that is too with AOCS, so I'm excited as well. As has been told, I'm going to talk today in this webinar about a part of my research which is being done at University of Concepcion in Chile as a part of my postdoctoral project. I'm handling this project with government of Chile since 2 years. And in this webinar, an overview of whatever work has been done in last 2 years will be presented. As the title suggests, my work is related with the biomass of a special type of yeast obtained from the Chilean rainforest for exploring its potential suitability for production of biodiesel. So let's begin. The theme of my work is on biodiesel production. So let's have a look at how the fuel properties of biodiesel are affected by the fatty acid composition. Biodiesel is an alternative to the petroleum-derived diesel fuel, and the fuel properties of biodiesel are directly dependent on the fatty acid profile or the composition of its source, which is nothing but the oil feedstock. As you are aware, the oil feedstock for biodiesel varies from vegetable oil to animal fats, or more recently, microbial oils such as algal oil or fungal lipids are also being used. In most of the processes, methanol is used for the production of biodiesel by trans-esterification processes. As a result, a mixture of fatty acid methyl esters is obtained, which we call biodiesel. So biodiesel is nothing but FAME or fatty acid methyl ester mixture. Now, the individual properties of these fatty acid methyl esters will ultimately determine or influence the individual fuel properties of biodiesel. However, the individual properties of fatty acid methyl esters are in turn determined by their structural features, such as chain length and degree of unsaturation. So in other words, we can say the biodiesel fuel properties are ultimately influenced by the chain length and degree of unsaturation of the constituent fatty acid methyl esters. Now, which are those properties which are influenced by the fatty acid composition? These include Sitter number and ultimately exhaust emissions, cold flow properties, viscosity, lubricity, etc. Now we will look how the saturation, unsaturation, chain length affects these different fuel properties. If the oil feedstock has a very high amount of saturated fatty acid, the resulting biodiesel will have a higher Sitter number. A good ignition quality is expected if the oil feedstock has high saturated fatty acid. It will also give oxidative stability to the fuel. However, too high saturated fatty acid will also result in poor cold flow properties. For this reason, a certain degree of unsaturation is required in oil feedstock. No doubt, unsaturation will give rise to a better cold flow properties required for cold climate. However, too high unsaturation might make the resulting biodiesel viscous, and it will also lower down the ignition quality by lowering down the C10 number. So, to balance the different fuel properties which are conflicting with each other, a special category of fatty acid is required, and it is nothing but monounsaturated fatty acid. For example, methyl oleate or methyl palmitoleate. These fatty acids are warranted being liquid at room temperature, and they impart higher C10 number without any adverse effect on other properties such as cold flow, and as well as they also impart good stability. So ideal biodiesel is made up of a synergistic mixture of, I will repeat, ideal biodiesel is made up of a synergistic mixture of saturated and unsaturated fatty acids. It is expected to have a single fatty acid uh, in a concentration as high as possible, and the single fatty acid should be monounsaturated such as methyl oleate. Moving towards the oleaginous yeast for biodiesel production. Now, why I selected oleaginous yeast for my research? The reasons are pretty clear, which are depicted in this slide. First of all, oleaginous yeast is nothing but a special category of a microorganism which has ability to store at least 20% of its dry biomass as lipid. The minimum lipid content is selected as 20% for the practical purposes of lipid production and extraction. The maximum lipid content can go as high as 70% naturally. Uh, why oleaginous yeast are suddenly becoming interesting candidates for biodiesel production? The initial interest in oleaginous yeast and filamentous fungi is credited to their ability to store or synthesize lipids which are not commonly found in plant or animal kingdom, such as cocoa butter or shea butter. So basically, yeast lipids or fungal lipids are not new to the alchemist. However, recent decade has seen interest in using yeast lipids for biodiesel or other types of biofuel production. The biotechnological advantages have made this possible. For example, they can be grown in a conventional bioreactor. We don't need photobioreactors or any special kind of reactor. Their growth, the growth of yeast is independent of light or climatic variations. Scale up of the process is easier. They have short life cycles, so growth because of the rapid growth rate. So the process is faster compared to the other microorganisms such as algae. The most important characteristic of oleaginous yeast is that they have ability to use lignocellulose-derived sugars, which makes possible the use of abundantly available lignocellulosic biomass, and ultimately it plays an important role in overall process economics of biomass to biofuel process. Now, if you look at the different unit operations or the process operations involved in using yeast biomass for biofuel production, this slide will try to explain uh, a different consideration is required when you look at oleaginous yeast for biodiesel production, uh, because oleaginous yeast has a rigid and robust cell wall. If you look at the cell wall chemistry of the oleaginous yeast, it has very high mannan and chitin content, which makes it resistant to cell lysis, resistant to pressure, and also resistant to pressure, which are the factors considered necessary for the cell lysis. Uh, the important step in lipid extraction or recovery from yeast is the cell lysis. Now, as it is rigid and robust, it is necessary to alter the cell wall permeability so as to facilitate the lipid extraction. A variety of biomass conditioning or cell conditioning methods are available in the literature which alter the cell wall permeability, and it ultimately leads to cell lysis required for lipid extraction. Physical biomass conditioning methods such as microwave, ultrasound, bead milling, high pressure homogenization are used. Chemical methods include the use of acids, alkali. Even enzymes are being used for the as a biomass conditioning method. At the end of different biomass conditioning methods, cell lysis takes place. Cell lysate is obtained. This cell lysate has extracted lipid, but for this we need to apply solvent extraction, liquid extraction. To the extract, lipid is extracted, so debris is separated. Then the lipid extract is subjected to washes with salt solutions to remove non-lipid contaminants. Basically, it is a purification stage, and at the end, you obtain the crude lipid extract. Solvent is evaporated, and it is subjected to further downstream processing or analysis by for the fatty acid composition studies. Now let me take you to Chile. Uh, as far as my research is concerned, the yeast on which I'm working has been isolated from the Valdivian temperate rainforest, which is present in southern cone of South America, as you can see on the map, which is almost at the end of the world. These unique rainforests are among the last 5 remaining temperate rainforests of the world. It is the second largest rainforest and the only rainforest in South America. The yeast has been isolated from the decayed wood of this rainforest known as palo padrito. It is a local Spanish word used in Chile. This decayed wood is used as a feed for cattle by the local farmers for more than 100 years. Palo padrido, or the decayed wood, is a phenomenon of wood degradation of woody substrates of native trees such as Nothofagus in this Valdivian temperate rainforest. In 2015, uh, uh, we went to this rainforest and obtained the samples of decayed wood to see whether this decayed wood can give us some yeast population involved in degradation processes, because the decayed wood phenomenon observed in Valdivian temperate rainforest involves white rot fungi followed by yeast population involved in advanced stages of degradation. As this wood has low nitrogen content, I hypothesize that this wood might harbor yeast which can accumulate high amount of oil or lipid, because lipid accumulation requires low nitrogen or nitrogen stress. So high carbon to nitrogen ratio is a prerequisite for lipid accumulation or storage in oleaginous yeast. So we obtained the decayed wood samples from the rainforest. We came to the laboratory and a number of yeasts were isolated. And one of the best yeast isolate was further taken. It was identified on the molecular sequencing methods as Schiphysomyces coipomoensis. And we found that it has an ability to store more than 20% of its dry biomass as lipids. We also found that nitrogen stress, metabolic stress by nitrogen limitation can induce the lipid storage in this yeast. And as the decayed wood in this forest have high amount of xylose, naturally this yeast has an ability to utilize pentose sugar, which is an important renewable resource for biomass to biodiesel production. So these are the important characteristics I found in this yeast, and it was further taken for the studies on biodiesel production. Now, here you can see the step-by-step fuel production from starting with the plate culture, which is grown in the laboratory. The first important step is to generate the yeast biomass, which is normally which at first stage are done at lab scale in shake flask or Lime flask. And as I told you, the lipid accumulation in yeast require metabolic stress, which is nothing but nitrogen limitation. So a cultivation medium with high carbon to nitrogen ratio is prepared. It is made with high concentration of D-xylose. If you can see the composition of the medium, it has a very high concentration of D-xylose supplemented with nitrogen in limitation in the form of yeast extract and ammonium chloride. So naturally the medium will provide high carbon in limitation of nitrogen, which will induce the lipid synthesis. The yeast were grown at 25 degrees Celsius. The temperature was optimized. The biomass was harvested by centrifugation. Then it was heat dried and pulverized and later subjected to formation of fatty acid methyl esters or biodiesel by in situ transesterification. This is a one-step methanolysis process which converts intracellular lipids directly into fatty acid methyl esters. The catalysis used for this is hydrochloric acid in presence of methanol and chloroform, which act as both solvent and reactant. The reaction has been optimized at laboratory scale. It took 20 hours and it had been carried out at 70 degrees Celsius. At the end of transesterification, the reaction mixture uh, is subjected to rotary evaporation of the solvent after separating the cell debris, and then fatty acid composition is analyzed by GSAFATI, that is gas chromatography coupled to flame ionization detector. So these are the results. The first important step in research was whether the yeast on which I'm working is having any lipid or not. So we did microscopy. Confocal microscopy was used by growing the yeast isolate, that is Cephasomyces coipomoensis, under nitrogen limitation. And as I told you, the yeast Oleaginous yeast store lipids as intracellular lipid bodies, which can be detected by a fluorescent probe such as nitrate, which has been done in this experiment. The first slide here is a differential interference contrast where you can see the intracellular lipid bodies. I'm pointing out with my cursor. The intracellular lipid bodies are clearly visible, which is nothing but the stored lipid. And the second image is the Nile red fluorescence with the confocal microscopy itself. And here you can clearly see the fluorescing intracellular lipid bodies. So it indicates that the yeast does have ability to store lipid as lipid bodies. Going to the now Nile red fluorescence has shown you the qualitative assessment of lipid accumulation, and it proved that yes, it does store lipids. Moving to quantitative, I did a time course of biomass and fatty acid methyl ester production up to 120 hours, and the results are in front of you. These are the preliminary studies in shake flask level. As you can see, the yeast is able to store up to 25% of its dry biomass in 48 hours. The lipid storage was found to decrease on further incubation, although the biomass tend to increase. So we can say under unoptimized condition, the yeast can store up to 25% of its dry biomass as oil. And the biodiesel concentration or the FAME concentration obtained was 1 gram per liter by 48 hours. This table shows the fatty acid composition by gas chromatography. The results clearly show that the lipids produced by yeast are enriched in monounsaturated fatty acid, more than 90%. And most of the monounsaturated fatty acids are dominated by these fatty acids. These are nothing but the oleate. Both cis and trans isomers of methyl oleate were detected. The possible reason of presence of trans isomer could be the elevated temperature and lengthy reaction involved in situ transesterification. Combined together, 73 to 75% of methyl oleate is obtained in the fatty acid methyl esters of the yeast oil. The second more prominent monounsaturated fatty acid was methyl palmitoleate. So to summarize, the yeast oil is enriched in both methyl oleate and methyl palmitoleate. Conclude, uh, Concluding the results, I would like to say the oleaginous yeast isolate Schistosomiasis coipomoensis has an ability to accumulate lipids using D-xylose. The lipids are enriched in monounsaturated fatty acids such as methyl oleate, methyl palmitoleate. It also has absence of linolenic acid as well as highly unsaturated long-chain fatty acid esters, which are the prerequisites for good quality biodiesel as per European specifications. So depending on the fatty acid composition, preliminary evaluation of fatty acid composition, we can clearly say that the lipids of Schistosomiasis coipomoensis has potential to produce biodiesel feedstock, and it is anticipated to have good fuel properties such as certain number, oxidative stability, as well as better cold flow properties. The importance of this study lies in the fact that yeast has ability to utilize D-xylose fraction, which remains the underutilized fraction of the lignocellulosic biomass. And there are very few um, yeast naturally present which has ability to utilize this fraction. You need to genetically modify them, but this yeast has the wild strain of yeast has this ability. My future studies are focused on increasing the productivity of these lipids by combinatorial approach of site-directed mutagenesis. Currently, I'm focusing my studies on mutagenesis of the wild strain to increase the productivity, which is also coupled to statistical optimization to improve the production. I would like to acknowledge the National Fund for Scientific and Technological Development of Government of Chile, that is Fondesit, which has funded my project, and my sponsor, Professor David Contreras from the Biotechnology Center of the University of Concepcion. And I also thank the Millennial Science Initiative by the Ministry of Economy, Development and Tourism of Government of Chile for constant encouragement for the research. And that's all. I thank everyone for your patience and attending this webinar. And I also thank AOCS for giving me this opportunity. You are free to hear the presentation again, we will have a chance to hear me again at the upcoming virtual annual meet, which is supposed to start next Monday. And you can reach me at the virtual annual meet. You can have a live chat, and yeah, that's all. Thank you.
Thank you, Dr. Coate. What a wonderful job. Very much appreciated.
Thank you so much.
Yeah, great job.
Yeah.
If you do have questions, please write those in the question box, and many of you already have, so we'll go ahead and get started with these. So our first question is, what is the potential for this technology to make industrial quantities of biofuels? World production of petroleum is a normal year, maybe not this year, is over 4 billion metric tons per year. Can you see being able to replace a significant portion of this? Diesel used for freight transport is currently of the order of hundreds of millions of tons per year. And I know that's a lot, so I can also push this over so that you can see it as well.
Yeah. So what I get from this question is whether we are able to fulfill the high demand of petroleum using the oleaginous yeast. Well, I would like to answer in a different way. This is an alternative feedstock, one of the feedstocks. Definitely, the whole petroleum cannot be replaced with any single type of alternative feedstock. Now, as there are different types of feedstocks are upcoming, algal feedstock is upcoming. Different types of fuels are also upcoming. So we have to keep looking for alternatives which can altogether replenish, or at least they can um, fulfill a fraction of the demand. And we have to start with the scratch. These are laboratory studies. Any industrial technology starts at laboratory, and I would like to say that at pilot level, yeast lipid based biodiesel production has been found to be sustainable and feasible. Many reports are existing. I'm not saying the yeast lipid will totally will replace the petroleum, but it has potential to replace a fraction, because lots of alternative biofuel technologies, biodiesel feedstocks are upcoming, and yeast is one of them. So I'm just stating the potential. It's the studies are still going on, but it does have potential. Yeah, I hope I answered it.
Yes, and if there's follow-up to that, Alan, please just write in another question and I'll grab it and add it to that so
Yeah,
that you get clarification.
the
Okay.
application.
Or if he needs clarification, I'll let you know and we'll go back to that question.
Yeah,
Um,
sure.
okay, the next question is, how did you decide what ratio C and N to keep in medium?
Okay. Which ratio or what ratio? What is the question?
I just shot it over to you so that you could read it as well.
Yeah. Yeah. What I did is I checked different combinations of CN ratio, and by doing statistical permutations and combinations, I zeroed down on this carbon to nitrogen ratio, and I found to be optimum to give the highest lipid accumulation under other unoptimized conditions. So basically, I first focused on CN ratio, different types of permutations, combinations were used, and then I zeroed down on it. So yeah.
Okay.
Hmm.
How is it better than algal biofuel?
Uh, I'm not presenting it as a competitive or as a competition to the algal fuels. I'm stating it again as an alternative. Mm, but if you compare it with algae, the advantage definitely lies in bioreactor, because algae do need photobioreactor most of the times. Um, but yeast can be grown in conventional microbial reactors. Whatever the existing infrastructure we have right now, because yeast are not only used in biofuels, they are being used for lots of industrial processes. So compared to algae, they are easy to scale up, they're easy to grow. Uh, the growth rate is faster. So if you compare with algae, they do have these advantages.
Okay, thank you. Next question is, why use chlorinated solvents?
Yeah. uh, these are lab scale studies. Uh, I'll be happy if chemistry will come up with the best organic solvent for chloroform. To the best of my knowledge, chloroform is the best organic solvent for lipid extraction, at least at lab scale studies. Yes, there are non-chlorinated solvents such as hexane or ionic liquids are also upcoming as good eco-friendly methods for extracting lipids. However, the scale-up of ionic liquids or other green solvents is quite complicated. So as of now, I'm restricting my studies at lab scale to the chlorinated solvents, but no doubt when the green solvents will become feasible, I will definitely use them, because that's also part of my research to replace chlorinated solvents with more eco-friendly, more green solvents. So these are unoptimized preliminary studies for these purposes, and these are lab scale. So and solvent recycling is being done as well. Yeah.
Okay. Our next question thanks you for the presentation and asks, please comment, is it possible to cultivate your yeast not only for biofuel, but also for the food industry as microbial or algal oil?
Yes, yes, there is a possibility, definitely. My project was focused on biodiesel, but as the research progresses, you realize many offshoots of your research. So definitely, oleic acid doesn't have only application in biodiesel. Uh, Also, for food industry or the other oleochemicals, for example, lubricants also, oleic acid is desirable. And if you look at the food industry, high oleic sunflower oils, high oleic safflower oils are also being used. So definitely it does have potential for non-fuel applications. What I presented today was focused on exploring the fuel potential depending on the fatty acid profile. You can play with the medium composition, you can play with the genes, which I'm doing right now through mutagenesis, and you can alter the fatty acid composition to make it suitable for any of application you desire. But in that case, if you want to use this yeast for food applications or non-fuel applications, you need to give more attention to the solvents which you use. You have to use food grade solvents. You cannot use chloroform. You have to use food grade solvents, because the total quality control changes when you use when intended use of the product is for human consumption. So principle remains the same, but the downstream thus varies. So yes, this yeast, I'm also considering this yeast to be used for non-fuel applications such as food. Thank you for this question.
Okay. Is high oleic oil of yeast edible?
It will be edible if you use food grade solvents, which I already answered in the previous question. I cannot use chloroform or other chlorinated solvents to extract the oil. If I use food grade solvents, then definitely this yeast oil would be edible. But if, given the fatty acid composition, yes, it is edible.
And this is just a follow-up question to a previous one, is a request to please repeat the exact ratio of C to end.
Exact ratio of carbon to nitrogen,
Correct.
it was 40.
Okay.
Yeah.
And if anyone has further questions, oh, I see one more that has just come in. Can make the breeding to this yeast to get high cis 18-1 and low trans 18-1.
Yes. Definitely. That's a drawback of in situ transcription, which I did. The process which I used uh, went on for 20 hours and at 70 degrees Celsius. So elevated temperature for a longer time normally is supposed to cause cis to trans isomer formation. So I had to play with temperature and the time of the in situ transcription, or I might have to shift to other transcription steps to minimize the trans isomer formation, uh, which is different objective for my project. But I will definitely look into it because trans is definitely not desirable, and it is also making the oil more viscous, which I found. But the work is underway to minimize the transformation.
Okay.
Yeah.
What is the oleic acid content provided per pound of residue or non-oil fatty acid remaining?
Can you repeat?
Yes. What is the oleic acid content provided per
Yeah.
pound of residue or non-oil fatty acid remaining?
Non-oil fatties. Basically, you should have a synergistic mixture of both saturated and unsaturated. There is no fixed amount of any particular fatty acid that it should be present in these. Because oil feedstocks are known to vary in fatty acid composition, but you have to look at the overall composition. And what I found is that above around 50 or above oleic acid is desirable in oil to have better fuel properties such as Cetane number. So you can say 50% of oleate is necessary, minimal. Yeah.
Please provide an example of a green solvent best for lipid extraction for the food industry.
Food industry. Uh, I think a number of ionic liquids are being used right now for the extraction. Also, the CTAB or some the switchable organic solvents, which are recently looked into, the switchable solvents, but these are still being used at laboratory level. Commercial level application of these green solvents, it is still far away, whatever the results are at laboratory level.
Okay.
Yeah.
And then we do have kind of some follow-up for discussion. And this is from Alan Payne. He's another one of our annual meeting
Yeah.
presenters.
Yeah.
And he's indicated that hexane is normally used for soybean extraction. And then he also had a follow-up discussion point that he wouldn't expect much cis to trans at the 70 degrees, that it was
Yeah.
perhaps caused by the catalyst.
The hydrochloric acid probably. Yeah. But also the time, because I'd read in the literature that the time of reaction that I carried out for 20 hours, so possibly. Yeah. And also I agree with the catalyst also plays a role.
Okay.
And thank you for the suggestions. Yeah, I will look into that.
Yeah, Alan's great and has offered to chat with you during the virtual meeting so that
Yeah,
kind of
I would be
collaborate around.
happy. Yeah,
Yeah.
That is the advantage of these webinars, and yeah, that's what virtual meet. So I'm looking forward. Thank you, Alan. Yeah.
Hey, another question is what would be its cost if its production is commercialized?
Cost, okay. That's difficult to answer. I'm not going into the feasibility studies or cost analysis. That's altogether different research. But I'm trying to cut down the cost. I mean, main aim of this process, the project is also to cut down the cost by using uh, lignocellulosic sugars, because fermentation substrate plays an important role in the overall process cost, followed by the solvents which you use. So to cut down the cost, I'm targeting the different lignocellulose-derived sugars, starting with D-xylose. But no, I haven't done the cost analysis. But once I scale up the process, it would be more feasible to do, not at laboratory level, because there are lots of limitations to do cost analysis. If I scale up the process at pilot plant, for example, say 1,000 liters, then I must do the cost analysis.
Yeah.
Yeah.
And that was the last question I see. I'll just give everybody a moment. Should they have another? Oh, and here they come. Jatropha is also the alternative for biodiesel. And what's your opinion on it?
Uh, on what? With?
Jatropha? I'll send it over so that you can see the question as written.
Yeah, just a minute. Jatropha, yeah. Uh, Jatropha, fine. Jatropha is one of the most talked about biodiesel production alternative. Theoretically, Jatropha does have a very high potential. The seeds have very high content of oil. It can be grown in arid land, and it's non-edible feedstock. Personally, I feel people went too fast to commercialize it. Uh, while the R&D was at nascent stage, people went to commercialize it without checking the feasibility studies, without checking the techno-economic analysis. That's why I feel it failed at that level. It does have potential, but we need more R&D into techno-economic analysis. And I personally feel uh, there are many socio-economical political roles to play. They come into picture when you deal with Jatropha. It's not about technological suitability of Jatropha. It's about you have to deal with farmers because the source of Jatropha is from the farmers. So you have to deal with the policies play an important role, and that's why Jatropha is not taking up. But scientifically, it does have potential. So all these alternative sources we have to work on together, which can displace, coming back to the first question, which can together displace a fraction of petroleum. No doubt, petroleum can never be replaced by any one single alternative fuel. You have to work on different types of biofuels, different types of feedstocks. Instead of pulling down the another feedstock, no, my feedstock is better than this one. You please use this. No, I would like to say we have to work together, all the alternative technologies, and which can at least displace, it will reduce the dependence on petroleum fuel. Even a 5% or 10%, it will make a big difference at environmental level and at economical level as well. That's what I feel. Yeah.
Yeah. And then we have, could some of the non-oil content be turned into oil using yeast? And this is in regard to Jatropha. I'll send it over to you as well.
Yeah. I, yeah. Could some of the oil content of jatropha be turned into oil using yeast. Yes, for example, yeah, I think you're suggesting me the uh, the leftover from the seed oil processing. Like in Chile, avocado oil is quite in high demand. When you extract avocado oil, the debris or the byproducts of this oil extraction mm, can be used as a fermentation substrate to grow the yeast. So definitely in the same way, the de-oiled cake or the de-oiled cake can be used as a substrate, and definitely we can use it for yeast oil production.
Okay.
But one point I would like to point out here, it does have some toxic metabolites, the de-oiled cake, which might inhibit the yeast growth if you want to utilize it for the as a substrate. So we need to check the um, chemical composition of the de-oiled cake or the non-oil content of jatropha, which the Alan is suggesting.
Okay.
Yeah.
We now have a question that our asker says is off the topic, but I think that's absolutely just fine. And they want to know a little bit more about your PhD journey as well as your fellowship, if you don't mind telling us a little bit about that.
Yeah, I wouldn't mind. My PhD journey, I completed my PhD in India from the University of Pune. Uh, However, at the end of my PhD, I was offered a visiting doctoral researcher place at University of Concepcion in Chile, where I'm currently doing my postdoc. At the same place, I came in 2015 as a visiting fellow, and that's how my collaboration with this university and the government of Chile started. I went back to India after a short stint in Chile in 2015. I worked for government of India for 2 years on East Lipid based biorefinery. I finished that project, but my mind was always in Chile. So, because I was interested in exploring the rainforest ecosystem, so I wrote a project to government of Chile to explore the more potential of the yeast isolated from the rainforest. And my first attempt failed in 2017. I applied again, and government funded that project in the second attempt for the 3 years. And I came back to Chile, and I'm happy to continue my research over here. So this fellowship is from directly from the government of Chile. Yeah, and I'm the principal investigator, and this COVID situation has definitely affected my work. But once the labs resume, I will continue with genetic modification and to improve the productivity, because as of now, the productivity is quite low. I'm able to increase it from 25% to 40% by statistical optimization. Now I'm going to increase further by mutagenesis and genetic modifications in the remaining time. Thank you, Sachin. I can see Sachin has asked this question. Yeah.
Hey, thank you for sharing that.
Yeah.
Another question we have is, has the oil of this yeast have antioxidants like tocopherols and phenolic compounds?
Has the oil of this yeast has antioxidants? Uh, no, the particular yeast on which I'm working doesn't have antioxidant, because normally the antioxidant properties are contributed by the pigments. There are other type of yeast such as red yeast known as rhodotorula or rhodosporidium, which has natural antioxidants in the form of carotenoids. But the yeast on which I'm working doesn't have carotenoids or any type of pigment. So probably I don't think it has any antioxidant properties. But the analysis can be done to like he has suggested tocopherols and phenolic compounds. So yeah, there is a possibility, but carotenoids are absent, so I don't think it has antioxidant contributed by carotenoids or any other pigments.
Thank
Yeah.
you. And that's currently all the questions I see in the queue.
Yeah.
Thank you, everyone, for such a great discussion and asking such great questions. And Dr. Coate, you've done a fantastic job following up and answering these. I will remind everybody that Dr. Coate will be presenting or have a live chat session next week on Thursday, July 2nd at the virtual AOCS annual meeting. So should you want to, after you've had a chance to digest all of this and follow up and have more time to chat, please join for that live chat session. And registration is free, so please join us. And with that, I'll thank Dr. Coate again for sharing this with us, and thank you everyone for attending.
Thank you, Amy. Thank you, AOCS, and thank you, everyone.
Have a great day, everyone.